Hello friends, welcome to Think Forward. This is Shrestha and today I'll talk about regulation of gene transcription in prokaryotes, and in this video I'll discuss about Lac Operon. So as we know, the Lac Operon is a model which was given by Jacob and Monod in 1961. In this model, what happens is that there is a short DNA segment. This is the short DNA strain in which different regulatory sequences are there from which different regulatory proteins are bind. For example, promoter is situated between the cap site and operator. Operator is present between promoter and these three structural genes. Okay, so there are two regulatory proteins. First one is cap and the second one is repressor. Okay, repressor binds to the operator and blocks the passage of RNA polymerase in this direction. So the transcription of these three structural genes cannot occur when repressor binds to the operator, and these three enzymes cannot be formed. Okay, when repressor binds to the operator, it blocks the passage of RNA polymerase. Here the RNA polymerase recognizes the promoter and binds with it and moves in this direction. So when it comes in contact with the repressor and comes in contact with the operator, it blocks its passage and the transcription of these three structural genes are inhibited, or we can say it is blocked. So the translation of these three genes cannot occur which are involved in the lactose metabolism. These cap, which is a catabolite activator protein, and Lac repressor are responsible for turning on or off the Lac Operon in response to the different glucose and lactose level. So for example, if we place E. coli cells in a culture containing glucose only, so two conditions arise. If glucose is present as the only carbon source and no lactose is there to bind with the repressor, the Lac Operon remains off, which means there is no transcription. Okay. So as we know, the Lac Operon is an inducible operon. The operon in which inducer is present to induce or increase the rate of transcription and to bind with the repressor is called inducible operon. Okay. So here inducer is lactose. Here, lactose acts as an inducer which binds with the repressor and prevents its binding with the operator. So here, repressor cannot bind to the operator and transcription of these three structural genes can occur, and resulting in the formation of these three enzymes after translation that results in the metabolism of lactose in bacteria. So here, lactose and its analogs like allolactose are very necessary for the transcription to be occur. So when we place E. coli cells in a culture containing glucose only, the Lac Operon is inhibited because cap is also acts at low levels of glucose. It binds with the cap site and promotes RNA polymerase binding, but only at the low levels of glucose. Oh, clear? And cap senses the glucose with the help of CAMP, which is a secondary messenger, that is cyclic adenosine monophosphate. So its function is to activate the transcription, but only at the low levels of glucose. So when glucose is present, cap does not bind to the cap site and transcriptional rate decreases. Okay. So the second condition is when glucose and lactose both are present, what happens is that lactose binds to the repressor and therefore the repressor cannot bind to the operator, and a small amount of mRNA is produced in this case. A small amount of mRNA is produced in this case, and what happens is that the transcription of the Lac genes, that is the three structural genes, and resulting in the formation of the enzymes are lifted, and the glucose is present, so the cap protein does not bind to the cap site in the Lac Operon. Thus the transcription is not that much lifted as in the case when glucose is absent. So at the different glucose and lactose level, the Lac Operon functions differently. So this is all for today. In the next video I'll talk about some other topic and other series. So stay tuned. Thank you so much.